My name is Clint Valentine. I am the VP of Data Science at TwinStrand. I've already been introduced, and I'm really happy to be here and share with you some of our technology. Like was mentioned, I have a background in genetic toxicology and then more so in software development. So I'm a little nervous for the heavy hitting medical questions at the end, and I might have to phone a friend. Just wanted to let you know that. So today we're going to talk about a couple things. They're all centered around ultra accurate next generation sequencing and their applications for AML, MRD, and cell therapy tracking. But first, we need to introduce the problem. And the problem is that when tracking really rare cells in, let's say, a patient or another system, we might use genetic variation as the biomarker. That would be a mutation. And there are challenges to detecting them when they are rare. So we need a method that can overcome that challenge. Detecting clonal genetic variants in NGS, like the two germline polymorphisms shown on the left, is relatively easy because the signal for a clonal variant is far above the limit of detection of your average DNA sequencing machine. If we were to search for a trace mutational signature of a rare cancer cell, for example, a cell at 1 part per 10,000, you may not be able to find evidence of it because its variance would be buried below a carpet of noise caused by the NGS assay itself. And you can see this here, where we have mutant fraction on the y-axis, homozygous variance at about 100%, so in every single genome and every single cell, heterozygous in half of the genomes, and a carpet of red noise. This can be caused by many different types of artifact generating processes involving sample handling, library prep, or the sequencing process itself. And what that looks like if you were to zoom in, if you were hunting for a very rare variant, is it wouldn't be possible to detect that rare variant against that background shown here on the right. The solution is to use an error-corrected NGS approach. That way we can drive the error rate down of the system to below the level of where we're going to detect our rare variants. We do this by leveraging the dual encoding of genomic sequence from the two complementary strands of DNA. This is mocking a real-world situation in which the cell uses both strands in order to error correct the DNA sequence itself. What we do is we start with a duplex-seq tag that you can see up here on the left. Inside this tag is a nucleic acid barcode that can uniquely label every single piece of DNA that we put into the reaction. And we do this in such a way that that barcode is asymmetric. There's some chirality within it such that we can determine the difference between a top strand of DNA and a bottom strand. Now, this is important because we want to track these molecules through the system. So later, when we're analyzing the data computationally, we can error correct out artifacts because we've labeled everything perfectly from the beginning. So I'll walk you through this figure briefly to show you how we can achieve the error correction without modifying the sequencing instrument at all. So we can use a base sequencer, of which there are many in the world, without necessarily modifying its protocol. We'll take a molecule with a, maybe the laser pointer is not a great idea. We'll take a molecule with a rare variant, so that's an AT mismatch. We will then amplify it up with PCR. This is before the sequencing process. Because we've tagged both strands of this molecule, we can put them into two different groups. So every single amplified product of the top strand there is shown on the top, and the bottom products are shown on the bottom. You can see that clonal variant exists now in all of the daughter copies, but what now appears are artifacts and errors introduced earlier through the process. We've got some that look like your random mistake. That could be a base calling artifact in the machine. But we have others like this clonal C down on the bottom right that could have been caused by an early round jackpot mistake that propagated to all of the other copies. That's a really difficult thing to overcome without having that top strand to use as reference. What we do after that is an informatics step in which we form a consensus sequence of these multiple molecules, and that eliminates nearly all of the errors. So the consensus sequence on the bottom would reveal there might be a T mutation or variant and a C mutation or variant, but on the top, after forming a consensus, there would only be an A. Knowing that that C does not form a consensus with the top strand, we would put an ambiguous placeholder there, and we wouldn't call it, which would mean that that artifact, it wouldn't be shown in your data. So this technology we call a duplex sequencing because it uses both strands, and it will help you pull back the curtain of low frequency noise so that you can see true variants. This is an anecdotal example of a region in the genome from an individual with a heme malignancy who was sequenced with both standard NGS and duplex sequencing. And what you can see is when we limit the variant allele fraction to 1%, we look for things that are ultra rare to this person. We don't see much. I mean, we see noise and we can't tell truth from artifact. But using an error-corrected approach, a true variant is revealed. And in this example, I mean, you can imagine that variant would be clinically actionable. It could be indicative of drug resistance per se, or it could be a variant that could be used for disease monitoring or tracking. Because what we do is basically extremely accurate NGS, it can be applied in many different fields. I'm going to talk about the two on the right today, which are more oncology and organ and cell transplant related. But these are all the areas in which duplex sequencing can have an impact. The company that I work for, we focus in the three that are colored. So genetic toxicology, everything in purple, which is a little more oncology related, and then in blue, organ and cell transplants. Let's first talk about residual disease monitoring in AML. So MRD status is the most outcome predictive biomarker in AML patients, and there's a need for more easy to use and accurate assays. In this figure, we show a few scenarios for clonal evolution. In red is a clone shrinking and then growing, but never to the point where it is undetectable by any standard assay, really. In purple is a clone which becomes even smaller. And if you had a limit of detection around 10 to the -4 in this figure, you wouldn't really be able to tell the practical difference between that clone and the one in the blue line until later monitoring time points, in which case the purple trajectory relapses or rebounds and the blue does not. So this is relevant for AML because as we're tracking that disease through time and clones are getting smaller, we want to understand better, are they truly dropping to zero? Are they lingering at some basal rate, or are they rebounding? And we would like to know as early as possible if that rebound were to occur. So we wanted to test the performance of duplex sequencing for this application. So we did a spike-in experiment to simulate a patient with minimal residual disease. The rarest variant in our spike-in has a variant allele frequency of 1 in 100,000. So it's well below the limit of detection of your base sequencing machine. With no error correction, it's not even possible to separate signal from noise. Every loci in these cancer genes appears mutated. We know that's not true, though. They're really just artifacts. And with simpler forms of error correction, a few of the more clonal spike invariants are detectable, but we have still that carpet of false positives. Moving down to a sort of duplex grade error correction, we were able to find all of the variants that we had spiked in, all the way down to 1 over 100,000 as far as a frequency goes. And this was a pretty robust proof of concept. One thing to note about this figure is that there's actually, we detected a few more variants than the ones that we had spiked in, but at extremely low rates. And it's very difficult to see here. But a lot of those, we think, are actually the background mutations of cells. Cells dividing, they're not perfect when they divide. There will always be some mutation rate inherent within the cellular processes. And these small blips that we see, we believe are actually that background mutation rate at maybe around 10 to the -9 for frequency. So to see a variant that you're not necessarily expecting doesn't necessarily mean that it is either an artifact or a clone that came with a disease, but it could be an age-associated process as well. So we've got a couple experiments. I'm going to show you a few highlights. This is an actual experiment, retrospective, in which we measured recurrent blood cancer variants at remission time points using a recurrent AML panel. I think this is a 29 gene cancer panel for AML, and these were 67 patients and individuals that we sampled. An early finding from this work revealed that the model that we built from duplex sequencing related data was more predictive to disease outcome than an existing gold standard assay for MRD. So this was really exciting for us. This was a first pass retrospective study with colleagues Jerry Radich, Megan Othis, Brent Wood, and Chris Horgan. And we're hoping to see this published very early next year. So something you can go and read about. I want to talk a little bit about designing panels for NGS and targeting disease-specific situations like AML, as well as a power detect based on how much clinical material you might have and what that means for tracking mutations. So in cases where clinical material is limited, how could you possibly increase your power for detecting a disease clone? There's only so many genomes you're putting into the reaction. Well, what we do is we will design a more broad panel in the patient. So we'll look for more markers, more genomic sites that could have an informative allele that we can then use for tracking. And by putting more sites in a panel, it increases our probability of detecting one biomarker if one were to exist. And we can get around sort of a lack of clinical material in some cases. This doesn't always work, of course, because those mutations need to eventually be there. And some diseases do not have very many mutations that are suitable for tracking. So in those situations, a static recurrent panel may not actually be a good fit, and you may need to take a more personalized approach. So this can be shown here, where to detect a given clone, we have a few variables we can tweak, and one of them might be fixed, which is how much DNA or input material we could put into the reaction. So generally speaking, if you have a very accurate technology, greater depth would equal greater sensitivity, but it always does require more DNA. So if we're limited by that, the only way to increase our probability of tracking a clone with genetic variants are by adding more sites in which those variants can occur to the panel. So that is shown here. Where recurrent panels or static fixed panels won't be fit for purpose is in cases like this, pediatric AML, where there are very few recurrent mutations. They might be private to that individual, not shared with necessarily any others. In those cases, we would go and design a personalized panel. This is a proof of concept study that we did with Ben Huang and Sohail of UCSF and Fred Hutch. We designed many patient-specific duplex sequencing panels and tracked clones through diagnosis, induction, and ultimately relapse in some scenarios. We found that duplex sequencing was very predictive at determining if a relapse would happen or not. But one interesting point about this study, and this is hopefully going to be published next year, is that the clonal dynamics that we saw, the dispersion of variants and clones at these ultra-rare levels, it was more complex than we had previously thought or understood. So in some situations, we saw those clones drop down during treatment and then come right back up and follow a very simple model of relapse. But in other cases, we saw dispersion of clones. And it's an interesting thought experiment to wonder if any of those dynamics or metrics could be perhaps informative in their own right. Could they be prognostic? So this was an interesting finding from that study. Taking the personalized panel approach, I'd like to move into another topic, which is how could you use this technology not only for tracking the patient's disease or cancer, but also for tracking allogeneic cell therapies or the therapeutic itself. In the gene editing and cell therapy space, rare variants can be used as polymorphic markers. So they might be rare in the therapeutic, but they could be baked into the carrier germline of whatever you're engineering. And this is specific to allogeneic cell therapies. But you can use those. You can use those as biomarkers for the persistence of that therapy. The challenge is, though, is that those therapies are probably persistent in that patient at a very, very rare level. So how could you detect them without an accurate technology? This is a challenging task, but something that we wanted to do a proof of concept experiment in. So to test our capability at tracking polymorphic clones in a CAR T therapy versus a carrier genome, we did a titration experiment. Now, this allowed us to reliably see the spiked in sample at rates below 1 in 1 million, which you can see here. And this set the stage for giving this an attempt in a retrospective study with 2 individuals that both received CAR T therapies. So here are those two individuals. The dashed lines represent the limit of detection of other available technologies. And we found something really quite interesting is that both these patients showed somewhat different dynamics. On the first scenario, we see a drop in abundance of that therapeutic detected by the assay, and then it comes back up and then levels off. And in the second example, we don't see that. We see a drop and then a leveling off. And it might be due somewhat to the granularity of the time points. But we're interested in some of the data that we're going to analyze in the future, where we'll have a better understanding of how these clonal dynamics last over time. So in conclusion, I hope I convinced you about the power of the technology and how it can overcome some of the detection limits of conventional forms of DNA sequencing. With duplex sequencing, individual variants existing at levels below 10 to the minus fifth can be detected, making this assay a very powerful tool for rare variant detection. We've shown that this tool can be used for recurrent adult AML MRD tracking, as well as patient-specific pediatric AML tracking. And we're also finding that as we sequence deeper with more accurate technology, that biology is proving to be a little more complex than previously imagined. But we're excited to learn more about that, because with some of this really crisp data, perhaps we could build more prognostic models based on what we're seeing. I think that's it. Yeah, so thank you for your time. Please reach out to me or TwinStrand if you'd like to learn more. I'm happy to take some questions about the technology. We have someone from our team coming in a little later today to say hi, but I'll be around all day and perhaps most of tomorrow. So I really appreciate you, and thank you.
So we're open for questions now. Ahmed?
Oh, thank you for this talk. My question is, have you tried or thinking of combining the MRD with chimerism testing? Because sometimes you cannot find specific markers for cancer, and in that case, you might rely more on chimerism. But with this high sensitivity technique that it can use chimerism with this sensitivity to detect cancer cells coming back.
Oh, chimerism. So I think that is ultimately what we are detecting through the tracking of some of these mutations, right? Did you mean it any other way?
Well, with chimerism, so this mutation is only in AML, right? But
yes.
Not in every types of hematological malignancy. So the other way that to do this is to do chimerism testing. But chimerism cannot detect if it's cancer cells coming back or if it's normal cells coming back.
Yeah, I think that's a fantastic idea. The panel that we have, it sort of balances cost of sequencing to a certain depth plus breadth, so that you can catch most of those recurrent markers in that disease AML. But conceivably, you could design a fit for purpose panel for any use. So you could go broad and target many more mutations. I mean, at some point, we'd like to envision a world in which we can sequence the whole exome or genome with data that is this accurate, but that is a little cost prohibitive at the moment.
Okay, thank you.
Oh, yeah. Oh, sorry, I'm just going to respond to Ahmed. Ahmed, I guess it also is relevant on what level of chimerism is clinically relevant, like detecting 1 in a million cells. Not sure.
Well, I think with chimerism, I think the clinician accept up to 5%. But if they have seen a trend of increase, like say maybe 0.1, and then the next 2 weeks it becomes 0.2, and then 0.3, and then they can actually detect relapse before it actually can be seen clinically. So it will be a nice tool as well.
Yeah, so we're 5% is quite high if you think about how many magnitudes lower we're able to reliably report what is happening in those frequency ranges. I think a new definition of MRD fit for the study would be needed, actually, because if you think about the replication error rates of the cells, if the deeper you sequence and the more cells you consume from a person, you could imagine a mutation at every residue in the genome. So at that point, what is MRD?
Thank you. I think we have a question. Oh,
I missed it, but can you do an HLA cross?
You probably could, actually. So the way that these, yeah, you could. No, I haven't tried it, but that's a good idea. The way that the technology works is you would have the adapters in a chemistry kit that you could ligate onto molecules, and then the panel is another variable. So you could change that. So you could have custom or hybrid panels where you're looking for these genes for this reason and these genes for another.
Oh, yeah.
Yeah, so it's a really interesting technology. How easily is it going to be dispersed beyond the site? So,
Yeah.
or do we have to send everything to Seattle? So
No, you can send stuff to us. We'll run it, but we do sell a biochemistry kit. So it's in a distributable kit format. If you have sequencing centers that you know of, they're probably capable of running biochemistry kits. And then the analysis is all done in a highly regulated cloud-based environment on the cloud. So freely distributable, we hope, to all sites.
And I have a question on the virtual chat from Jia Huai from VGH, who would like to ask, how many % of the AML MRD cases does this assay apply to?
Yeah, that's a great question. So the panel that I showed you today, it's a 29 gene panel. And I think we estimate, depending on how you get this number, that it should catch about one mutation in around 93-ish% of adult AML patients. There are ways of getting that number a little higher that we're looking into, but they broaden the panel substantially. So there is a cost trade-off there. And there are diminishing returns because some of those final variants that you want to use as a predictive biomarker, they show up in fewer and fewer proportion of the diseased population. In that case, AML, we're a little lucky in that the 29 genes does a pretty good job of catching most adult cases. But like I showed with the pediatric cases or even cancers that do not have many recurrent variants, you would probably pursue a personalized approach where you may sequence at time of diagnosis, determine which markers you'd like to go after, print a panel, combine it with a duplex sequencing kit, and then you would be able to do MRD tracking specific to that individual.
Thanks, Clint. Are there any other questions? Oh, one more question. How long is the turnaround time?
How long is the turnaround time? I think you can get it complete in about a week, including library prep and analysis, around there, plus or minus.
Are there any other questions? Oh, one question, sir.
Sorry, I'm just wondering how this overcomes like the conventional constraints around the centromeric and telomeric type genes. Like it seems like it would have the same pitfalls as like normal sequencing.
Yeah,
short read.
That's a really good question. We're overcoming sort of the random mistakes that are happening in amplification, PCR, sequencing itself. However, regions of the genome that are highly repetitive, they're just going to be tricky anyway. And it may be even more of a computational problem, but certainly a sequencing problem too. Everything I showed you today was based off of the Illumina short read sequencing platform. Short reads do have a weakness in that if you've got really, really long repeats or highly repetitive regions of the genome, you're going to struggle to map and align reads into them. So that would still be difficult.
Oh, sorry.
I think I saw, did I see one more question up there? No, just me hallucinating. Okay, thank you so much. Thank you, Clint.
Thank you.